Good morning to everyone. So I would like to thank the organizers to giving me the opportunity to present our work. And after having talking about eliciting immune responses, we are talking about unwanted immunogenicity. So today I will talk to you about the impact of therapeutic protein aggregates on the activation of dendritic cells. So my talk will be divided into two parts. As said Jeremy, I will expose you some results that we obtained in the Paris consortium, and then I will talk about our work currently done in our lab. So, as you may all know, patients that are treated with therapeutic proteins may elicit some unwanted immune responses, and they may develop the so-called anti-drug antibodies, ADA. And this patient can get immunized in two ways. First, they can get immunized following a break in immune tolerance, which is the case when they are administered with the recombinant form of endogenous proteins, such as factor VIII coagulation, or for example, erythropoietin. Or a patient can also get immunized with foreign epitopes, which is more the case with monoclonal antibodies. So this ADA production would lead to the bioproduct neutralization with the loss of biological activity, but there may be also some adverse events that, for example, are allergic reactions, such as with the cetuximab treatment. So many factors can influence immunogenicity, and among them patient and treatment-related factors, but also, and here we are interested about product-related factors, and precisely the presence of aggregates in the product that is administered. So aggregates can form during the production process, but I will say that at this stage this is very controlled, and they are avoided. This is no more the case when handling or administrating the product, because then the product may undergo some stresses that are uncontrolled, and aggregates may form at this time. So aggregates may form, for example, when two monomers that are partly unfolded will interact to form first a reversible interaction, but they can also make an irreversible interaction to form a small nucleus that is the first small aggregate. And then this aggregate will grow and to give bigger particles. So the presence of aggregates and immunogenicity have been related mainly using transgenic mice models, in which mice were tolerant for the study of human native proteins. And among these studies, they can be shown that, for example, for interference, aggregates can induce an immune response. And also this has been shown for antibody aggregates. So the hypotheses about cellular mechanisms involved in ADA production are quite classical. The therapeutic proteins is captured by antigen-presenting cells, such as dendritic cells. And then the hypothesis is that aggregates may play as a danger signal and activate dendritic cells that are in turn able to activate naive CD4 T-cells. Then these T-cells may help B-cell maturation and the isotypic switch to finally give the development of ADA of high affinity. So in this context, dendritic cells play a pivotal role in the establishment of this response. In the literature, you will find some in vitro mechanisms that have been shown, especially PBMC activation with antibody aggregates and T-cell proliferation, and probably the receptor engaged here are TLR or Fc receptor, gamma receptors. Also, there are a few publications about aggregates that will activate dendritic cells, and but there are very few papers about that. So first, I will talk about the work we performed in the Paris consortium, and our work package was in charge to develop some in vitro assay for the prediction of therapeutic antibodies immunogenicity. And precisely, we performed some standardized DC maturation assay, and together with 4 industrial partners, we done these studies to compare 4 different in vitro assays. So the antibodies that have been studied here are antibodies that are prescribed for inflammation diseases. And it is natalizumab, rituximab, adalimumab, and infliximab. So we first prepared 4 batches of aggregates, and these antibodies were submitted to heat stress with 2 intensities, and also to shear stress with also 2 intensities. Then aggregates preparation were characterized with orthogonal analytical techniques, and here are the results that are summarized for the heat stress, because the heat stress gave more propensity to aggregation. And here you can see that infliximab at the naked eye showed the highest susceptibility to heat stress, and then it was rituximab, whereas adalimumab showed moderate susceptibility to heat stress, and natalizumab showed the lowest susceptibility. Then we use these aggregates to test DC activation in a monocyte-derived dendritic cell model. So in short, we took some blood from healthy donors, and independently the 4 labs prepared the dendritic cells. The PBMC were isolated, and then the monocytes were purified using magnetic CD14 beads. And then monocytes were cultured in the presence of GM-CSF and IL-4, and that will give some immature dendritic cells. So this stage of immaturity was well controlled, and then immature DCs were incubated either with native or aggregated antibodies, and we took some positive controls that were maturation cocktail with cytokines or LPS. And then independently, the 4 labs perform one assay. Here the surface activation markers exploration with a cytometry method. The quantification of chemokine and cytokine mRNA expression with quantitative PCR, or also the quantification of the cytokine and chemokine release with a multiplex assay. And then we started to explore the cell signaling inside the stimulated disease with Luminex assay. So here are the results for the phenotypic experiments. They are expressed as the fold increase of the expression of surface activation markers. As you can see here, I will focus on the infliximab compared to natalizumab. And you can see here that neither native antibodies nor shear stress aggregates were able to upregulate these markers, whereas for infliximab you have upregulation of CD83, CD86, and not CD18. But with natalizumab, there was only a slight increase in about CD80. The conclusion is that infliximab induced more significant activation with this phenotype study. Also, we next quantified the production of cytokine and chemokines. As you can see here, for infliximab, heat stress aggregates were again able to induce significant upregulation of pro-inflammatory cytokines and also CCL3 and CCL4 that are chemokines that induce T-cell recruitment. And there was also, of course, a nice correlation between these results and the mRNA transcript quantification. On the other hand, you can see that natalizumab aggregates were not able to produce cytokines or chemokines upregulation. Only here a slight increase in CCL3 production. So to go further on the cellular mechanisms involved in DC activation, we explored some signaling pathways with Luminex multiplex assay. And again, here we can see that for infliximab aggregates, heat stress aggregates, there was upregulation of the phosphorylation of SYK, AKT, and ERK1/2 that was significant. Whereas with natalizumab, there was no significant increase of these phosphorylations. So to sum up this first part, we observed significant hemodc activation with infliximab aggregates, which was the antibody that showed the best propensity to form aggregates. We tested 4 different assays, and they all go in the same way. And we can say that infliximab aggregates induce a DC1 phenotype, that is to say, a pro-inflammatory DC phenotype that may drive T-cell activation. In contrast, we observed much less activation with natalizumab treatment, and I didn't show the results, but with adalimumab and rituximab, we had middle variation of all these parameters. For example, only one phenotype upregulation, or only one phosphorylation for one intracellular protein, for example. So we can say, in seeing the aggregation propensity, that the results we found are in line with this aggregation propensity, and also they are in line with the observed immunogenicity in the clinic, where infliximab show much patients with ADA development than natalizumab. So moving to the second part of my talk, I will present you here some results about a second model that is a human growth hormone. And why human growth hormone? Because it's a simple recombinant protein, unglycosylated and quite small, 22 kilodalton. And an old publication showed that patients that were treated with different formulation preparation developed the more antibodies where there were more aggregates in the preparation. Fortunately, now the patients that show anti-drug antibodies for this recombinant protein, the antibodies are not neutralizing antibodies. So we took this example and generated some aggregates by inducing a shaking stress to this protein. And then the aggregates were characterized with several orthogonal methods. With size exclusion chromatography, we were able to show that 46% of the protein was aggregated as insoluble aggregates, and there were very little dimeric or multimeric species. With dynamic light scattering, we were able to show one main population at about 900 nanometers. And with morphogranulometry, we were able to show that there were bigger particles ranging from 1 to 5 micrometers. And also we performed some fluorescence spectroscopy and circular dichroism to confirm that mainly all the protein was in a denatured manner. So in the first paper, we were able to show that these growth hormone aggregates were able to modulate in the same model of dendritic cells, monocyte-derived dendritic cells. Aggregates were able to upregulate the phenotypic markers CD83, CD18, 86, and also MHC class II molecule. And these activated dendritic cells showed also a signature of cytokines, secreted cytokines. And here you have the whole signature, and you can see that this is a pro-inflammatory signature with here the upregulation of CXL10 that will recruit T cells and IL-12 P40, and to show that these activated dendritic cells were able to induce further specific immunogenicity. We performed some co-cultures with allogenic T cells. And we were able to show that these CD4 T-cells were able to proliferate and to mostly secrete interferon gamma, which is a Th1 profile. Now we've started also to explore the signaling pathways that are involved in the DC activation by this growth hormone aggregates. And as you can see in this picture for Western blots, you can see that here the main MAP kinases can be phosphorylated. Here P38, ERK, and JANK, and also the subunit NF-kappa-B P65 subunit that is also phosphorylated. And by a second method, we could show that this subunit can also bind to DNA probes, meaning that NF-kappa-B can translocate into the nucleus. We also, we also use specific inhibitors of these pathways, and we show that IL-12P14 and CXL10 mRNA expression is also under the control of these MAP kinases and also NF-kappa B. We wanted to test the possible recruitment of other intracellular proteins that are implicated here in purple, that are Rho GTPases, that are implicated in the control of acting networks during phagocytosis. And also we explored PA3 kinase involvement. So again, using specific inhibitors of those Rho GTPases, we were able to show that the production of IL-12P14 and CXL10 that were induced by aggregates could be inhibited by all these inhibitors. So we can see that Rho GTPases and PA3 kinase are implicated in the production of these proteins. And finally, using the same inhibitors, we showed here that Rho GTPases are also implicated in MAP kinases P38 and ERK activation, here and here, but not PI3 kinase. And JNK is also dependent on PI3 kinase. So to sum up this part, this signaling study, we were able to describe some intracellular pathways that are implicated in this, is stimulated by aggregates of growth hormone. First, the recruitment of proteins that are implicated in phagocytosis, and then MAP kinases that were able to promote the secretion of cytokines and also the expression of phenotypic markers. And then we are currently working on the identification of internalization pathways of these aggregates, and also we are working on the identification of the receptors that could be involved in aggregates recognition. So in conclusion of our work, we can say that you see that therapeutic proteins show very variable susceptibility to stress. We were able to study different readouts, to identify different readouts of this immaturation, and to show that this model is a valuable tool to assess the intracellular mechanism that drive DC activation. And also with a study with antibodies, we were able to show that this signal may recruit FC receptors with the recruitment of intracellular SYK. So as a conclusion, we can conclude that there is one signal, signal one, that is the impact of aggregates on DC maturation that can be predictive of immunogenic potential. But we can also ask if the stability of a therapeutic protein is also relevant as the aggregate content in the drug product. And we can also ask what is the real part of aggregates induction in vivo behavior. And the second signal that I'm not talking about today because it is well explored in the Aggregates consortium, is the recognition of aggregates and also bioproducts by T-cells. And for that, there exists a large toolbox that is developed in the case of the consortium. For example, in silico studies, HLA binding assays, MAP assay or T-cell assays that we already talked about yesterday and maybe later. So to finish, I will like to thank all the people that participated to those works, especially Dendritic cell team, and very especially the PhD students that perform all the tests. And also I would like to thank people that help us for the characterization of aggregates, and also industrial partners that we were working with in the Paris consortium. So thank you for your attention.